Hello, everyone. Thank you. Thank you, Tatiana, for the introduction. And wasting no time, I'm moving on the presentation. In non-scientific literature, but also in taxonomic or phylogenetic papers, we can find many terms regarding structures on the surface of insect's body. These are terms such as bristles, hete, sete, spines, spinous tubercles, pilosity, pubescence, and hairs. But of course, the term hairs refers only to the structures that grow from fossils found in the dermis of mammals, which constitute an autapomorphy for this group of animals. Most of these structures are sensory organs, sensilla. And here we face the first problem: how to distinguish sensilla from other cuticular structures. Theoretically, it is simple. Sensillum is characterized by having at least one neuron that is absent in non-sensory organs. Unfortunately, checking this requires sophisticated methods and advanced light microscopy or electron microscopy. Its dissemination took place in the 70s of 20th century, and one of the first studies focused on Miridae trichobothria. The method that facilitated and popularized the research of sensory organs is scanning electron microscopy. Okay. A suitable scanning microscope allows determining the function of individual sensilla based on their morphological features. The advantage of this method is that the preparation of material for testing in CIM is relatively simple. Functionally, there are three types of sensilla: mechanoreceptors, chemoreceptors, and Thermo and Hygroreceptors. The same sensillum is usually responsible for temperature and humidity perception. The morphological division, on the other hand, is much more complicated. Researchers use various, usually incompatible classification. For example, this sensillum, we can find a whole of terms in the literature, such as sensilla basiconica, multiporous placodea sensilla, multiporous olfactory sensilla, or sensilla trichoidea, and others. But it is not everything. Many subtypes are distinguished, indicated by letters, numbers, or shape names. The terminology of sensilla definitely needs to be revised. In addition, some taxa have sensilla with very characteristic shapes not found in other insects, e.g. in waterbugs, leaf-shaped, club-shaped, brush-shaped sensilla, and other unknown. But the information provided in this presentation will be only about terrestrial true bug. Most of the large sensilla on the insect's body are mechanoreceptors. These receptors can perceive mechanical stimuli such as touch, sound, vibration of air, water, and ground, as well tension of bending the cuticle. So they are responsible for the sense of touch, hearing, and proprioception. Cuticular mechanoreceptors can be divided into two types: protruding hair-like sensilla with flexible socket, and campaniform sensillum characterized by presence of dome instead of appendage. Flexible sockets allow for deflecting sensilla, which stimulates the dendrite types. Hair-like mechanoreceptors can be modified in various ways, e.g. flattened or shortened. And a quite spectacular example of mechanoreceptor modifications are spinous tubercles occurring in the inner surface of the Metaphomora in Femurocoris, New Caledonia representative of Miridae. Perhaps the most recognizable type of sensiloop in Heteroptera is Trichobothrium. It's trich situated in a cup-like depression in the cuticle, bothrium, or a small dome surrounds of its base. Trich is fine and long, everything almost along their entire length, flexible and tapered apically. Their surface is characterized by parallel grooves. Bothrium is frequently surrounded by trichome, an area of densely arranged microtricha. Despite many characteristic features, Trichobothria shows considerable diversity. For example, on the Miridae femora, we can find those with a typical structure as well as reduced ones. The number and arrangement of Trichobothria are highly conservative, and therefore they are important in the diagnostic of family group taxa and phylogenetic studies based on morphology. The diversity of Trichobothria positions on the body in various taxa of Heteroptera could indicate that these sensilla have different origins. The Trichobothria are located on the head, antennae, as in the case of Reduvidae, scutellum, labium, and abdomen, for example, in Legaoidea. And in the Miridae, 3 to 8, rarely more, the Trichobothria are located on the lateral and ventral surfaces of the meso and meta femora. This is an important diagnostic feature of this family. In 2030, Liu Sigvald and Cheho observed a pair cephalic Trichobothria in the immature stages as well as in the imago of Fulvius Carioni. In the conducted studies, I have demonstrated that the presence of ventral cephalic Trichobothria is a characteristic feature of plant bugs. A pair of ventral cephalic Trichobothria was observed in all examined species. Each Trichobothrium of this pair is located laterally to the first article of rostrum on the gula between the buccula and the antennal tubercle. Moreover, a dorsal cephalic Trichobothria was observed for the first time. They are located above the antennal tubercle towards the center of the frons. Chemoreceptors are responsible for the senses of taste and smell in insects. The olfactory receptors detect gaseous chemicals, and the taste receptors detect substances dissolved in water, even if in dry surfaces. To detect chemical substances in the air, the sensillum must have pores on its surface through which molecules can enter to interior, where the dendritic ends are located. The olfactory receptors are found on the antennae and the labium. Taste sensing contact chemoreceptors usually have only one pore of the apex of the appendage, which is usually hair or peck-shaped. Taste receptors are located on the tip of labium. The olfactory receptors on antennae can take various shapes. There are often trichoid, but also hetica, basiconica, or celoconica sensilla. Unlike mechanoreceptors, the chemoreceptors usually adhere to surface of the antennae. As I mentioned, these sensilla have numerous pores, which are visible only with high magnification. The pores range from 10 to 25 nanometers in diameter, and their distribution varies depending on the type of sensory organ. Sometimes the same sensillum has a dual function, being both mechano and chemoreceptors. I will present chemosensory organs on the tip of the labium on example of neibids. Nevertheless, preliminary research results on Miridae indicate that they have very similar chemoreceptors organizations on the labium. In addition to the mechanoreceptors, numerous marked yellow and a pair of orange, there are 2 and sometimes 3 types of chemoreceptors in the labium. Marked with a light blue color, PEC sensilla with a terminal pore are responsible for the sense of taste. Elongated placoid sensilla with wall pores, red, and oval plate sensilla with terminal pore, brown, are olfactory organs. In addition, between the gustatory organs, there are 4 sensilla that function as Thermo and Hygroreception, marked in dark blue. It is noteworthy that the number of chemoreceptors and Thermo and Hygroreceptors appears to be a conservative feature in all studied Navidae, Miridae, as well as individual species from several other families have the same numerical scheme of this sensilla. Apart from the Thermo and Hygroreceptors of the labium, which I have just mentioned, the sensilla responsible for these senses are also located on the antennae. The most common are sensilla coeloconica, a non-pore PEC inserted into a pit. This type of sensillum is found in small numbers, and the mechanism of action of hygroreceptors is poorly understood. Probably changes in air humidity affect the water content in the sensillum cuticle, causing its deformation. Of course, not all structures on the body of true bugs are sensory organs. The hair-like appendages are assigned a covering and protective function. However, the role of many structures is unknown. Example includes thread-like structures described by Ibrahim and colleagues, or the cuticular circular formations we observe on both on antennae and the legs of Miridae. There is also a secretory pore system on the antennae, often associated with the basis of the olfactory sensilla. And this one is also interesting, whatever it is. I have seen a similar structure in mites. It was referred to as trichobothria, but I have any idea what is this. And an interesting issue is teratological cases of the antennae. Antennal anomalies can originate in the nymphal instars through injuries to the antennae and their subsequent regeneration during the next instars. Antennal anomalies in Pentatomomorpha have been observed much more frequently than in other infraorders. For example, in a study conducted on Aradidae, we found malformation of antennae in 2.9 of the cases. However, within the new species of Meschia, family Meschidae and superfamily Legaoidea, as many as 8 out of 33 specimens, for 24%, were characterized by antennae teratology. Oligomere, otherwise called symphasomere, which depends on reducing the number of segments that results from injury and the anomaly that has most often been reported. Interestingly, although the number of segments is smaller, they are larger than normal and the characteristic set of sensory organs is reproduced. This phenomenon is known as compensatory regeneration. Maintenance of sensory functions of the teratological antennae may be helpful in better understanding the functioning of the sensory organs. And conclusions. Most of the microstructures on the surface of true bug's body are sensory organs. Diversity of sensilla is certainly greater than previously assumed. There are cuticular structures whose function is completely unknown. And determining the homology of sensory structures and their phylogenetic and taxonomic significance requires further detailed ultramorphological studies that should include a wide range of taxa. I want to extend my sincere thanks to everyone who contributed to the results presented today, especially thanks to Adrian, who spent hundreds of hours with me working on CIM, and Professor Jolanta Brożek, who introduced me to the subject of sensory organs a few years ago. And by the way, if you know any species with unusual antennae morphology or with interesting structures, I would be grateful for the information. Thank you for attention.
Thank you very much.